All right, so it's a Friday in February. That means it's time for a journal club. We only have one more of these journal clubs. And in fact, I have so much good stuff to tell you about today. We actually have two parts to this journal club, two different papers we're going to talk about. One of them really sets up the other, and they're both very interesting and important, and they're both from Mark Davis's group. So you might detect a theme. We did Mark Davis's paper for the last one. So for this one, we have one about a fairly old paper about virus-specific CD4 positive memory-phenotype T cells, so T helpers, are abundant in unexposed adults. Now you kind of are going along normally until you hit that last word, unexposed adults. How do you have virus-specific T cells in an adult who's never seen the virus? Well, we'll explain. It has to do with cross-reactivity. And cross-reactivity is a fascinating subject, and it's a very complicated subject, and so it's important to take our time and to go through it in the right way. Cross-reactivity is one of the basic causes behind autoimmunity, because when you have a T cell that's not supposed to react with self, T cell is supposed to react with something other. It's supposed to react with a pathogen. And yet, instead of reacting with the pathogen, you have a T cell that's reacting with self. That's called cross-reactivity. It's crossing over to react with self, and this is one major cause of autoimmunity. Now, autoimmunity, because T cells can activate the innate immune system, and you can actually have cross-reactivity coming the other way as well, the innate immune system, as always, can be involved as well. And in fact, with autoimmunity, you don't just have this one kind of autoimmunity. There are general autoimmunities where there's not one antigen you can trace it to, but it's a general activation of the immune system that ends up being a general activation against all things, including yourself. Basically, autoimmunity means you have an imbalance where you're attacking yourself in one way or another. And there's an activation of B and T cells where you don't want them to be activated. And it can happen, we've talked about thymic selection, negative selection. If a T cell gets through negative selection and it still self-reacts, then you have a failure of negative selection. Now there's also the T cells are supposed to be put in sleep mode, anergic state. If that doesn't happen right, then they'll react with self. If you have self-reactive T cells, you want to turn them off. And if you don't turn them off, you have autoimmunity. Treg cells are supposed to turn the other cells off. If you have a problem with making those, then you have autoimmunity. And if you have certain programs that will delete self-reactive mature cells that are outside the thymus in the periphery, anything about that can cause autoimmunity, and also problems in antigen presentation. Going back to the beginning of the process, that can cause autoimmunity as well. So autoimmunity is like allergy, but it's different. Just because, like we talked about in class, how it's like cancer is involved with autoimmunity, but it's different because cancer is kind of other. Cancer is something you want to destroy, whereas yourself, you don't want to destroy it, right? So we got to be very precise in how we talk about things. Allergies are where you're overreacting to something from outside that you shouldn't overreact to. Autoimmunity is where you're overreacting to yourself. There is an overlap, though. TH cells are almost always involved because they amplify a response, and so if you have a problem with them, you have an improperly amplified response. And you might have organ-specific symptoms where the autoimmunity is against one organ, type 1 diabetes, pancreas. And systemic, it would be where you're more going throughout the whole system. Remember, we talked about lupus is sort of autoimmunity against your own antibodies. You have antibodies everywhere. IgG is going to be everywhere. And so lupus can be a systemic disease. Rheumatoid arthritis, by the way, is really more systemic, even though it shows up in the joints. I may have said that before, and that might imply that it's organ specific, but connective tissue is not really an organ. It's more everywhere. And so there are antibody effector functions. There's also damage reactions that can be part of the pathology. And the weird thing about autoimmunity is more women than men are affected. This is important because we're going to see we're going to see this showing up later in the journal club. So the disease does tend to be chronic and progressive. That means it's always there at a low level. And you may have a relapse where you get better for a while. You may remit it. One of my favorite authors, Flannery O'Connor, her entire life was changed when she got a lupus diagnosis. And eventually she died from complications related to that, I believe. And so autoimmune diseases are a big deal. You know, they hurt a lot of people. We can figure them out at some level. Anything we can figure out can help us move forward, you know, can help to heal people. So usually the treatment for the autoimmune disease has historically not been that specific. They're very complicated. The immune system's complicated. And then the immune system attacking yourself, you're a complicated person. And so that's two complicated things coming together. So what you usually do is you give anti-inflammatories, NSAIDs, you can read it, NSAIDs, right? You can give basically aspirin and things like that to try to calm down the immune system. And you can have general anti-inflammatories or immunosuppressive drugs. There are certain steroids that are immunosuppressive. You give them for asthma. You can also give them for autoimmune disease. Statins are another category that will just generally turn the whole body's response down. There's something called plasmapheresis, which is where you deliberately remove the Igs from the blood. If you have a problem with too many or self-reactive Igs, this could be one way to treat it. But you can see you're losing all the good they do as well as the bad they're doing. As we go more and more, we're getting to specific drugs. But honestly, I'm going to show you something that we just figured out that may lead to a specific drug someday. And by the way, our research project involves KIR receptors, which means we're going to talk about those having a role in autoimmune disease right today. So who knows? Any part of the immune system can be a potential lever if it turns out to be a specific lever for one specific disease. So the specific way to target, the easiest way to specifically target one part of the immune system is to make a monoclonal antibody. And in fact, I should look at actually I've got the chart right here. You know the chart that we had that was about the different the monoclonal antibody nomenclature. I'm just going to see, there is an immunomodulating category. In fact, our test question had an answer for which you decoded the name and you found out that it was an immunomodulating antibody because it had an L in the middle, and that goes with immunomodulating. That kind of antibody can be the kind of thing that you can give. It means it binds something in the immune system and modulates it, and that could be one of the examples of what it can do. Now, monoclonal antibody can also specifically bind to and deplete a T cell or a B cell. It can block activation. If you have an anti-CD3 antibody, it will block all T cells, and the anti-CD4 will block only helper T cells. Or you can block a co-stimulatory. Actually, those look the same. The T cell activation, there could be other co-stimulators that could be like that. And you can do certain things like there's a receptor. I'm not even going to say its name because we haven't mentioned it before, but it can inhibit the circulation. Basically, if you have a molecule that does something specific, an antibody can specifically block it. And then there's anti-TNF, where you have an antibody that binds TNF alpha and prevents it from causing its inflammatory results. Of course, these are very expensive. To get a whole antibody, they're hard to make, they're big, they're unwieldy, you have to make them perfectly pretty much for them to work. And so it's very expensive, and you have to keep administering it because you need the antibody to be around to continually block the thing it's blocking. But here's a news story that just came out the other day. There's a monoclonal antibody against lupus, and it's actually in phase 3, which is pretty well along the cycle. Maybe this will be one of the things we could decode the name, obinutuzumab. But it's probably better to call it Gaziva. I'm not sure. Anyways, whatever this is just an anti-CD something. Anti-CD20, it turns out that if you bind CD20, you deplete B cells. And so they give this to deplete B cells. That makes sense in lupus because it's an antibody autoimmune disorder. So B cells are almost certainly involved and overactivated. So what gives you an autoimmune disease? It can be a lot of times genetics. For example, MHC will involve autoimmunity. You're involved, if you have a particular MHC haplotype, you'll be presenting certain antigens and making certain kinds of T cells. If you have a MHC that presents a peptide that looks just like a self-peptide, then you will have self-reactive T cells, things like that. There are environmental factors that can trigger this. Again, this is overactive immune system, so maybe things that can make your immune system overactive, the typical diet, smoking, immunogens, microbes can do this, maybe even stress. And it is the gender puzzle. Why do women tend to show more vigorous immune response to pathogens? The one thing I want to say is maybe female autoimmunity is the flip side of male immunodeficiencies. Remember, we had the X-linked immunodeficiency that showed up in the boy that we talked about, the case study a few case studies back. It seems to me, this is just actually a guess from just seeing that. But I know that female autoimmunity is real. I know that male immunodeficiency is real. So maybe it's related to the fact that if you have two X chromosomes, you're more likely to have a more active and more reactive immune system. See what you think when we get to the end of the journal club, because that's going to have a little more about this. But this journal club is more about molecular mimicry, because remember that you can have infections that will trigger a disease. A lot of autoimmune sufferers report that they get sick, they get an infection, and then their autoimmunity flares up. This seems to be more than just a coincidence. This could be, so why is this? Well, it could be that just the immunity by itself just increases your inflammatory level, and you have a self-reactive T cell that's sitting around, and it's a bystander, but you have an infection that causes immunity inflammation to go on. And remember, that sort of activates the whole area. Maybe it activates that self-reactive T cell and it starts going after the self. That's definitely a possibility. But then there's also a more specific molecular explanation. Maybe a pathogen looks to the immune system like it is a self. Maybe you have a pathogen peptide that will look like a self peptide that's presented. So when you get that pathogen, you present that peptide, your T cells fight off the pathogen, but then they are on alert against that peptide. And it turns out that you have your own peptide, which looks exactly like that peptide to the T cells. This is the cross-reactive. And you have your effector cells expand, and then you have your T cells attacking yourself as mistaken identity. Then there's also a possibility that if you have inflammation or tissue damage, it might cause self-antigens to be presented more. Maybe if you have cells that are broken up, they'll be taken up by macrophages and then presented on the surface of the macrophages or dendritic cells. And so you can have just the kind of necrosis that might happen. Apoptosis hopefully packs up things so that they don't explode all over the place. But if you have necrosis, you can have antigens from that necrosis that get picked up and presented. And that is more of a problem with the necrosis, but it leads to autoimmunity. This is a case of what molecular mimicry looks like. You don't need to know what every single amino acid here. Every peptide is a short chain of amino acids. Each amino acid is represented by a letter. So if you have letters that are the same, then you have amino acids that are the same, and therefore you have peptides that are the same. Not just look the same, but are the same for those exact letters. And so look at this right here. You have human cytomegalovirus, IE2, has this sequence with the blue letters in the middle. And it's a short sequence, and this just happens to be the peptide that's in the virus that gets presented by the immune system. Well, some people have an HLA-DR molecule that has a peptide that's exactly like that in the middle, the part where the T cell sees. So after you get infected with cytomegalovirus, your T cells are looking around for this LGRPD sequence. It turns out that LGRPD sequence, there's only so many amino acids and there's only so many spaces. By chance, you're going to accidentally get something that looks exactly the same. And in the middle looks exactly the same. If the T cell reacts to that LGRPD, it's going to say, oh no, I found a virus. When it really didn't find a virus, it found an HLA-DR molecule snippet. But how can it tell? They're exactly the same. Poliovirus looks like an acetylcholine receptor. Papillomavirus looks like an insulin receptor sequence. Rabies virus looks like an insulin receptor sequence. There's all sorts of examples of these. So you can see that it comes down to the letters, and you can understand why the T cells are confused. And remember, this is more likely to happen with T cells because T cells only look at a 10 residue snippet. In fact, they only look at one side of the peptide, even better, because one side of the peptide is buried, right? So only one side of the peptide is pointed toward them. And the T cells basically will just by chance, these virus peptides will look like your own proteins, and the mistaken identity is going to happen. Just remember how T cells work. We've seen all this before. Remember the window or the hot dog bun? The peptide is just a little hot dog in the hot dog bun. And the hot dog is only like 10 residues, 10 amino acids long. And only 5 of those are pointing up. So it's very possible that those 5, just by chance, will be presented and it will overlap with what a virus peptide has. Remember the side view of these, that we have MHC class 2 and class 1. In both of them, the peptide is like the hot dog in the hot dog bun. And remember that there's some pointing down, some side chains pointing down, some side chains pointing up. The T cell can only see the things that are pointing up at it. You know, these peptides are going, the side chains are going up and down. They don't have a controlled up or down, and so that means they're half down, right? That's the idea, and that means that a T cell really is recognizing only a few peptide side chains that are pointing up toward it. By chance, that can happen to be shared, just like if you have a simple name, by chance someone else can have that name. I knew there were 3 Derek Smiths in my high school. And that's not going to happen for a more complicated name like McFarland. So just remember that we have all of these things. And so this molecular mimicry can activate you against self. But it actually cross-reactivity, that means you're cross-reacting between the virus and the self molecule. Cross-reactivity can actually work the other way. And the way they found this out kind of by accident. They found out this, and this is Mark Davis's lab. This is our journal, Club A. So after being exposed to some viruses, you'll have T cells that will react to other viruses that you've never seen. In fact, somebody who's never been exposed to a virus still has a chance of having a T cell that will react with that virus. And that means that something else is cross-reacting with the new virus. Something old has been activated in the past, and it's being activated in the present against a new thing, just because it happens to share part of that sequence. The molecules are mimicking. This is the way that it was shown. They were working with HIV reactive T cells, and they took a bunch of people, and they found that there were T cells taken from people who never had HIV that would react with and these are TCR clones, by the way, so they know exactly what the TCR is. So they're taking these TCRs from people who have never seen HIV, or who have seen HIV, sorry, and they found out that sometimes you'd see somebody that would react, a T cell from somebody who's never seen A. tropicalis. It would be reacting with A. tropicalis. They'd never been off the continent, and A. tropicalis is only on another continent. They could never have seen this. And they see this over and over again for different ones. Now, it's not every clone. Only some clones will cross-react, some clones won't. But you have a lot of weird peptides here, and yet you have 5 TCR clones that are supposed to be recognizing HIV and maybe other things. There's a lot of other things that they're recognizing, from weird bacteria, and I think these are all bacteria. Some of them might be viruses as well. And I wonder if this is related to children, if your T cells develop and they're always sick because they have T cells that don't cross-react. Maybe you're less sick because you have T cells that have been educated to be able to cross-react to new stuff. But immunity is complicated. This is just one way that this might show up. Now, I never knew exactly how this came up until I read this paper, which has got a great title, Standing on the Shoulders of Mice by Mark Davis. Mark Davis's argument in this whole paper is that we should be looking at human immunology more than we look at mice. And yet we're looking at mice because they're easy to manipulate. And yet there are significant differences between mouse and human immune systems. In the context of this argument, he mentions how they actually came up with this study before. So he's saying human studies are an ideal complement to mice. He's not saying that we shouldn't look at mice. He's just saying that we should re-emphasize human, even though it's hard. And why are humans different from mice? Well, we have genetic diversity that inbred mice lack, and we're also continuously exposed to infectious diseases that we avoid scrupulously in modern mouse facilities. We keep things simple and sterile in the lab. We are not living in a lab. We're in a complicated system, and we are more complicated organisms. So one of the examples that he brings up is that when his researcher asked, what was the pre-immune frequency of pathogen-specific CD8 positive T cells that might be in blood bank donors? So basically, they took blood bank donors and they had a new tetramer staining enrichment method. Tetramers are very useful tools for immunity. They allowed us to see T cells we couldn't see before. But they were quantifying. That allowed us to quantitate for the first time. How many T cells do you have? And they were asking, what is the immune frequency before you're infected by something? So they took a control and they analyzed for self-specific T cells, and they found, to their surprise, that the self-specific T cells were present and were very frequently found. You found a bunch of self-specific T cells, and then you found pathogen-specific T cells as well. And so this is like you're finding T cells that react to everything. We thought self-specific T cells were supposed to be removed, and yet they found not a huge amount, but a significant number of self-reactive T cells. That's because they got a tool that allowed them to look more clearly, and we found out that no, it's not that self-reactive T cells are completely absent. Negative selection still happens, but some of the T cells get through. Some self-reactive T cells get through. If that happens, there must be a reason for it. So the study went on to show this very clearly. Flu vaccination will present certain things against flu peptides, T cells against flu peptides. And there are other bacteria that have peptides that look similar to the flu peptide. So they wanted to see, okay, if you're immune to this flu peptide, we think that these other bacteria have peptides that look like the flu peptide. And look down there, you can see they all have a similar EKMNTQ sequence in the middle of it. So they said, let's take these and let's see if these T cells that were raised against flu, that never saw F magna anywhere, and yet will they mistakenly, or maybe on purpose, react with this peptide because it has the same middle, even if it has different ends. Lo and behold, they did. And it worked for one donor, it worked for another peptide from another completely different organism. Basically, the immune system is deliberately messy, and only 2 or 3 side chains really form strong contacts to the TCR. There are other ones that can be kind of medium contacts, but let's say that half of the side chains can even point up to the TCR. And so that means that it's pretty easy for the peptides to accidentally get KMNTQ. There's only so many letters in the alphabet. And so just by random, you're going to get a KMNTQ somewhere else. Then the T cell sees that, it will react to it. That's the important thing about cross-reactivity. The possibilities of life are limited by the number of amino acids we have. We only have so many letters. And because they're limited, by chance, you will get the exact same situation, the exact same structure in 2 different organisms. This can be a good thing. And this can explain why T cells can react. T cells that have never seen COVID, they'll definitely, when COVID came on the path, we definitely said this is a new virus. We've never seen it before. And that means that most of our T cells won't react against it, and we need to build immunity against it, therefore vaccine. But there was something that I said, I wondered from the beginning, what is the extent of cross-reactivity? Because we've never seen COVID-19 before, but we have seen coronaviruses before. And by chance, we could have seen something totally different. that happens to present a similar reactive peptide. Now, this is not enough on its own. I want to be very clear from the beginning. But there is some ways in which it might help. Something that is novel is never truly novel. That does not mean your immune system will not react against it at all. There will be a low level of activity that hopefully you can build on. And this is why you can actually get, before we had the COVID vaccine, we saw evidence that other vaccines did have some sort of protective event against COVID. This is an article about it. It's about how the trivalent MMR vaccine and the Tdap vaccine antigens, those particular vaccines, would actually give you T cells that would react with COVID. And they saw that it looked like if you had these two different vaccines before, you were protected, you were reduced somewhat against the severity of COVID-19. Almost certainly didn't affect infection. We've got to realize what the limits of the immune system are. But it definitely, according to this study, having the other vaccine, the MMR vaccine will actually reduce COVID severity. How much is this? This definitely is not as good a protection as an actual targeted COVID vaccine. But because the immune system is messy and it overlaps, there is at least some hope that we have some kind of reaction to even something that is completely new that we haven't seen before. Which leads us to the question of what goes on with these cross-reactive cells and why do we have them? That's going to be the next topic for the next one.